Thank you, Mr. Chairman. Good afternoon, everyone. My lecture will be about embryo selection and possibility of laboratory improvements. First of all, I'm a commercial guy, and so here are my collaborations with Ribogenetics and Life Global. And I may mention some of their data in the slides that are following. The points that I like to cover are, first of all, is embryo selection desired? That sounds like a silly question, but I want to go over it because some people have argued that it may not be. Then I'll continue with introducing a new technology. What does it take? What does usually happen? It follows a certain pattern, such as we've seen with PGS and time-lapse. Then I'll go to the new platforms of PGS, and particularly like to introduce it by considering all the debates that have occurred over the last 10 or 15 years, and particularly what the proponents have to say and the opponents have to say. And they actually have a lot more in common than they think. So I'll talk about that, and then continue on the safety of biopsy and the advantages of particularly trophectoderm biopsy in the last few years. And then, when there's a few minutes left, talk about the evidence that now exists in PGS. It's a work in progress. Of course, there are a lot of embryo selection methods, and only a few of those are listed here. These are probably the ones that are the ones that have been most progressed. And the ones I'll cover are in the middle, PGS and time-lapse. There are many papers about embryo selection, over 1,000. Can't list them all on one page, and I apologize if you don't see your name here. But these are the papers that have introduced these subjects the first time. And so many of these methods are, as I say, work in progress, and I think there's not a single one here that is a finished product. So first of all, the question is embryo selection desired? That would not be a question that one would ask if one is a technology lover like I am, and I presume most of you are as well. If you're a technology lover, you don't transfer an embryo without looking at it or without doing a test that exists now. Even if it's very simple, you would do that before you transfer. However, there are now some colleagues who argue that there is evidence accumulating that all available embryos in an IVF cycle can be cryopreserved and transferred in subsequent cycles without impairing pregnancy rate, or maybe even with an improvement in pregnancy rates, and survival rates are so high they approach 100%. This is a paper from Amsterdam University by Wong, Rapping, and Mastenbroek just a few months ago. And that argument is fine, but then they say embryo selection will then no longer be able to improve the live birth rate in IVF. It could even lower the live birth rate. Now, if you wonder about that statement, I refer to this as Dutch math, and I think I can say that because I grew up in Holland, and so I know a little bit about Dutch math. So if you want to get to 10 in the quickest way, and you have to use several numbers, 5 plus 5 is 10 would do it, or 2 plus 2, etc., is 10 would do it too. And so the argument here is, well, take your time, freeze all the embryos, and just transfer one at a time, and you don't have to do any testing. And then you get to 10 as well. And that's true. The endpoint is the same. It's 10. But the second pass takes much longer and is very painful. For anyone who has done IVF, they will tell you that it's a lot more painful and a lot more involved. So it's not a very simple question of mass. It's much more complicated. Which brings me to the introduction of new embryo selection technology and the new introduction of technology in general. And I've taken the example of time-lapse here. And this graph, many of you will have seen before. It's called the Gartner hype cycle. It basically is a response to a technology trigger, usually in electronics. Gartner is a company in Connecticut, and they look at the introduction and the continuation of the production of instruments and technologies, and do this for analysts. I call this the Gardner-Palmer cycle because it was Charles Palmer from Greece, who unfortunately is not here, who introduced this concept to biomedical research and particularly introduced it to IVF technology. And so what happens here, you have basically about 5 stages. First of all, you get a technology trigger, then you get a peak of inflated expectations. That doesn't happen in all cases. For instance, in eshre it didn't. In eshre there was expectations and they were not inflated, they were realistic. And then there is disappointment because of results, of series of observations, and it could also be randomized clinical trials. You then go to what Gardner calls the trough of disillusionment, and then a second and third generation of product developments and reassessments of the tools can cause a second and even a third slope of enlightenment. And then finally you get productivity. And so this is kind of what happened in PGS. And some argue this is also what's happening in time-lapse. And I only agree to a point. In time-lapse, this paper is from Armstrong and co-workers and was published this year. It's a paper on time-lapse. It's a well-written paper and thoughtful. How should we assess potential benefit? And the authors here in yellow argue, in particular, we emphasize the ethical aspects of hastily adopting a costly technology without current high-level evidence of improved live birth rates, safety, and cost effectiveness. Yeah, we all agree with that. However, it is a narrow view. It is narrow because time-lapse is done or should be done for a number of reasons. Only the last option listed here is about improvement of implantation, improvement of pregnancy rate, a better selection tool. There are other aims as well that are very profound. Quality management tool. You have a permanent data record when you do time-lapse. You don't worry about it as long as you back up the data. You have a permanent data record. You can always go back to it a few years later or even 10 years later. You don't have to do a changeover of embryos. Now, others have done that already before. But time-lapse brought that message home. If you don't do a changeover, there are less disturbances, and it seems to actually improve things. It's a proficiency tool, so you can standardize embryologist observations from one person to the next. And because you can do that within a lab, you can also do it across laboratories. So you can standardize across laboratories. It's also a research tool. And the last point, only the last point, is the improvement of implantation. So it's a narrow view to wait for evidence-based medicine to tell us whether these instruments should be placed in laboratories. I think they should be placed in laboratories if you can afford it. So this is, again, the Gardner-Palmer hype cycle. And in the case of time-lapse, there is a second slope, which is the progressive use cycle that's unrelated to pregnancy rate and has to do really with a better control and standardization of laboratory quality control. So this brings me to PGS. What do proponents and opponents have in common? Well, they have 5 things in common, 5 arguments in common. First of all, they all agree that many embryos are genetically abnormal. There is no doubt about that. There are hundreds of papers about it. Most of these abnormalities are spontaneous. Embryonic aneuploidy is maternal age dependent. Don't have to show you the curves. You know them very well. Aneuploidy and embryonic morphology are poorly correlated. They're correlated, but the correlations are not very strong. And we also see this in time-lapse studies. They're not very profound. And then finally, a big problem is that you don't look only at meiotic anomalies, you have to look at the post-fertilization anomalies as well, because mosaicism is very common, and both proponents and opponents agree on that. So these are the issues. Then another issue is that there's so much new genetic testing technology being introduced that it's hard to keep up. And so there are a number of platforms. This is a scorecard made by Dr. Mark Hughes from Genesis Genetics, which I've borrowed from him. And basically, this shows you the 5 technologies, and there are actually a few more, listed horizontally here on the top, with next generation sequencing on the right, and the older FISH technology on the left. The number of signals, which you can see increases, of course, with the sophistication of the new platforms, and then all the advantages that people are looking for, such as low false negatives, low false positives, simplicity for the embryologist. Can you do chromosomes and gene analysis all at the same time? And so what you're looking for in this particular scorecard is green dots, And the green dots are about the same in all the new novel technologies that have been developed in the last 4 or 5 years. But the one that is clearly marked with a star is next generation sequencing, because it gives you the highest number of signals and it gives you more data. So therefore more possibilities. So what about safety of biopsy? Well, it's a very, very big topic, and it's probably the single most important cause of why it has taken 15 and even 20 years for the field of PGD, PGS to develop. The first papers describing that biopsy of early cleaved embryos was potentially damaging came from Australia, from David Edgar in Melbourne, and the first publication was in 2000, basically correlating cell loss after freezing as indirect evidence for the loss of cells during biopsy. Transfer of biopsied embryos without PGS was performed by Dr. Mastenbrook's team in 2007 during their massive trial on the use of fish in cleaved embryos. And they found that these biopsied embryos had significantly lower implantation than the controlled embryos. So these were embryos that did not have PGS, they didn't have a genetic test. Well, there was a genetic test done, but there was no result. But the most compelling evidence comes from a small but very important study from Richard Scott, Nathan Treff, and others from New Jersey. They transferred one biopsied and one intact embryo, followed by fingerprinting. It's a complicated paper, and I advise you to read it if you're interested in this subject. They basically took patients who volunteered for this study, and they had transfers either on day 3 or day 5, depending on the judgment of the doctors as well as the patients. And then 2 embryos were selected, the best embryos, and they were randomized to either being biopsied or not in each group. And then no PGS was performed, no PGD was performed. The DNA was kept, and the DNA was matched by fingerprinting to the fetus if there was a fetus. And so the implantation rates of these fingerprinted embryos could be determined. And therefore you would know whether the embryo was biopsied or not. And so you can see this listed here. On day 3, the implantation rate after biopsy, which is BX, was 30%, and no biopsy was 50%, and that was a significant difference. And when it was done on day 5, there was no significant difference in implantation between biopsied trophectoderm biopsied embryos and those that were not. So obviously that's an important finding, even though some criticism exists about the way that was done, because the day 3 embryos were grown to day 5, were not grown to day 5, they were transferred, and the implantation rate of the no biopsied embryos on day 3 was relatively high, yet they were randomized, and therefore that's just a function of randomization. So what are the advantages of trophectoderm biopsy compared to cleaved embryos? Well, there are quite a few. More cells are biopsied, therefore there's more DNA, and the geneticists love that because they need that to make their tests more robust, more reliable. There's a reduced learning curve. That is, embryologists who know cleaved embryo biopsy and do biopsy of trophectoderm argue that they prefer trophectoderm biopsy because they get to the endpoint faster, and it seems to be easier to accomplish it. There's a lower error rate. There's a higher call rate. There is prolonged culture because you go to the blastocyst. It means that you lose abnormal embryos just spontaneously because of the fact that you culture them longer. That means you have less embryos to process and less samples to process. It reduces the cost, it reduces the amount of work. And of course, this combined, all these factors combined, give you the most preferred way to do single embryo transfer. So why didn't we do this before? Because the method was introduced by Anna Vega and Mireia Sandalinas and others in 1997. So it's almost 20 years ago. It was done in Barcelona for the first time. Why wasn't it done massively after that? It took another 10 years before the Genea team in Australia picked up on it, and then it took even more years for it to move to Europe and North America. So why wasn't this done before? And it was because we needed an improvement in culture system. And those improvements came slowly and then more determinedly in the last 5 to 10 years. So we now get much more blastocysts. We get better blastocysts. They have more cells. They have better cavities. they have a much better hatching rate. The chromosome testing platforms improve. So improved chromosome testing was a factor. And of course, vitrification became so successful that almost all of the embryos now survive, if not all of them. And so that combination made it possible. So what's the evidence? Well, evidence-based medicine, first of all, gives a green light to blastocyst transfer. So that's a good point already to take into account. The successful trials using tofactoderm biopsy are few, but they all showed kind of the same result, and it's a positive result. And so far, no detrimental outcomes reported. We have to keep our fingers crossed because there are other trials that are ongoing and more data is in progress with unpublished outcomes. So it's possible that there will be some negative results in the future as well. And so the findings of these trials and other work is that there are lower error rates, higher call rates, there's much more information, fixation is less variable because all you need to do is stick the biopsy like PGD in a PCR tube. That's all you need to do. Not that that is so simple, it takes a little bit to learn this, but it is very doable. It's much less invasive. And we see the result of these considerations in the way samples are sent to Reprogenetics in North America. In the blue bars here on the right, so on the horizontal axis, you see the number of cases done in 2002 and the number of cases done on 2014 on the right. And these blue bars represent array CGH cases, and the green are the old fish cases. There are hardly any fish cases left. Most of it is now done with array CGH or next generation sequencing. The light blue bars represent tofactoderm biopsy, and the darker blue bars still the day 3 biopsy. So you see a trend towards 24 chromosome assessment. You see a trend towards biopsy being done at the blastocyst stage. And so it's expected from this curve that it will be full 100% probably within 3 to 5 years. All of these cases will be done at the blastocyst stage. So what are the results of the randomized clinical trials? As I said, there are only 3, and they're listed here. And you see the control blastocyst implantation in this column, and the blastocyst implantation after PGS in this column, and all 3 showed a significant improvement. A modest improvement, but yet still significant. Of course, it's very easy to poke holes in these trials and argue that they weren't done properly. And I agree with that. I can poke holes in them too, and I can do that with the old randomized clinical trials as well, and I think you can do too. So it's a matter of continuing this type of work. This is a first result, and it's going in the right direction. It's not perfect by any means. There have been 2 systematic reviews published. This one from the previous speakers group, Dadu and co-workers, and they found higher implantation and ongoing pregnancy after PGS, and they said it improved embryo selection and elective single embryo transfer, but they said it's the same ongoing pregnancy, but significantly reduced miscarriage, and they were a little disappointed because it's only so far these trials were done in good prognosis patients. So certainly the old approach of doing this in advanced maternal age needs to be picked up again. And we need to look at, for instance, failure after IVF or multiple failures, whether such patients will be good candidates for PGS. Another review was published a few months earlier from Australia, and this review had basically the same conclusions, promising results in good prognosis patients, but they said there were only matched cohort studies in complicated groups. So particularly in the latter, more work needs to be done. So in conclusion, progress is technology. You can't progress in this field if you don't apply new technology. You need to do time-lapse. You need to do any of the other embryo selection methods. You need to do, I think you need to do PGS, so consider it seriously, maybe starting in a subgroup and then slowly, once you get experience, slowly expanding that if you have positive results. So does time-lapse need evidence-based medicine? I think it does for the one question that's asked about pregnancy, but not for the other reasons. Tofactoderm biopsy seems safer, In the second generation PGS, there has been now some success and some evidence, but we need more. I think if we apply Gardner's hype curve, hype cycle to PGS, we've gone through the peak. That was 10 years ago, with Master and Book here on the top, and the peak following and the decline. And I think we are out of the disillusionment and towards elimination. I think we're on the slope up with the second and third generation of platforms. What's going to happen in the near future with PGS? Blastocelic aspiration, first done by Dr. Pellini in Bologna, and very recently this year, results from Dr. Generoli, also from a group in Bologna, showing that there's a rate of 98% concordance when you do blastocyst biopsy and compare this to blastocele aspiration. So you can actually take fluid out of the cavity and then correlate that with trovectoderm biopsy, and it gives a 98% concordance rate. Maybe that's not as good yet as when you do 2 biopsies, but it's getting very close. So this is very exciting work, and it's considered non-invasive. Whether it's really truly invasive, it's a matter of debate, but it is an interesting step. Another application pioneered by Dr. Fraguli, Elpida Fraguli, and Dagan Wells from Oxford, is a correlation between the number of mitochondrial DNAs and therefore the number of mitochondria in early embryos with implantation failure. That can be combined, that can be assayed in next generation sequencing. So you can look at chromosomes, you can look at genes, and you can count mitochondrial DNA if you're interested. You can also look at mutations of mitochondrial DNA. And finally, I like the following colleagues for their kind assistance and suggestions over the years to come to these conclusions. Of course, none of this work, it's important to improve implantation, it's important to improve pregnancy, but the most important thing is the safety of the embryo and the safety of the mother and the safety of the healthy baby. And therefore, it's the next speaker who's going to cover that subject. Thank you.